The second of the special senses that I want to consider in this series is hearing. It might seem strange at first, but we need to remember that hearing is a mechanical sense. As we can see in this first slide, when we strike the tines of a tuning fork, that actually causes the air molecules to vibrate, and they move, setting up a wave. And it's the impact of that sound wave, the movement of the air molecules against the auditory system, that we detect as sound. The frequency of the sound, the pitch, how high or low it is, depends on the frequency of movement of the molecules, the frequency of the wave, and obviously the volume depends on the amplitude, the extent of agitation of the molecules. The sensory system, the auditory system, is incredibly sensitive. We can detect pressure changes of around 20 micropascals, which are absolutely tiny. After all, if you think of the sound of a clock ticking, the actual physical movement of the molecules of the air is going to be really small. And so the sensory system, our sense of hearing, is incredibly sensitive. So how does it work? When sound waves reach the ear, they're channeled via the outer ear canal to the first physical structure, which is the eardrum, or tympanum. The eardrum is a very thin membrane that's stretched tightly across the end of the ear canal, and the sound waves cause the eardrum to vibrate at the same frequency. Now, that's very easy when we have a single tone, as for example with the tuning fork, but most sounds are highly complex, a whole mix of different frequencies. Think of listening to an orchestral concert or a ranger conversation at a party. There's a whole range of different frequencies. So the pattern of vibration of the eardrum is very complex, and that pattern is disaggregated and interpreted within the auditory system. And that creates the sense of sound and our ability to identify different sounds. The eardrum forms the boundary between the outer ear and the next chamber, which is the middle ear. This is an air-filled chamber that contains 3 tiny bones, the so-called auditory ossicles. These are called the incus, malleus and stapes, or hammer, anvil and stirrup. And these bones play a very key part in the transmission of the sound from the eardrum to where the sense organ for hearing is located itself, which is the inner ear. The inner ear is fluid filled. So the sound has to be conveyed, the sound waves have to be conveyed from air to a fluid interface. Now, if you've ever put your head underwater in a swimming pool, you can see the problem. As soon as you put your head underwater, all the sounds that you could hear from outside tend to be muffled and confused. And that's because most of the sound waves are reflected off the surface of the water. They're not transmitted through the water. The sound loses a very significant amount of energy in passing from the air to the water across that interface. The role of the ossicles is to act as a lever system that mechanically amplifies the range of movement to compensate for that impedance of the transmission across the interface. Also, the surface area of the eardrum is much larger than the surface area of the so-called window that connects the middle ear to the inner ear, and that also focuses the pressure of the sound waves. So there is a mechanical processing, mechanical amplification of the sound that allows transmission with minimal loss from the air to the fluid interface. Once we get into the inner ear, what's happening is the ossicles move in and out in frequency response to the sound wave, and that sets up a new wave within the fluid of the inner ear. And the inner ear is where we find the sense organ itself. It's within a structure called the cochlea. And the cochlea is a spiral containing 3 chambers, in the middle of which is located the sense organ itself, called the organ of Corti. And this sits on a structure called the basilar membrane. Now, the basilar membrane has very important mechanical properties that are associated with the transmission and detection of the sound. The next slide shows the basilar membrane within the cochlea as if it had been unrolled. So, as I said, the cochlea is a spiral, so normally the basilar membrane is curved round. But what I've shown it here is it's unrolled in a straight line. And the basilar membrane changes in its physical properties along its length. Next to the oval window, which is where the ossicles first connect, the basilar membrane is narrow and stiff, and as we pass along its length to the far end, it becomes wider and floppier. And so its frequency response changes. So where it's narrow and stiff, it's sensitive, it will respond to high frequencies. At the far end, it responds to low frequencies. Of course, this is happening over a very small length. The basilar membrane in your ear is about 35 millimeters in length. The frequency response of the basilar membrane depends on its stiffness over its length, ranging from 10 Hertz at one end to 20 kilohertz at the other. It's a remarkable range. Not only that, but within that range, we have a very high acuity. So we can detect differences of a few cycles per second, a few hertz, between different sounds. So if you're listening to sounds, you can detect differences of pitch that are incredibly small. And this is all down to the mechanical properties of the basilar membrane and some associated neural properties which allow refinement of its acuity. So how are the oscillations of the basilar membrane converted into neural signals? So within the organ of Corti, as we can see in the next slide, are some groups of hair cells. There's 2 types of hair cell. There's the outer hair cells. So there's 3 rows of outer hair cells. And these play an important part in modifying the acuity, modifying the frequency precision of the basilar membrane. And then there are the inner hair cells, which are the main hair cells that respond, provide our sense of hearing, that respond to the frequency changes. So, as I said before, the ossicles within the middle ear move in and out in response to the vibrations of the tympanum, of the eardrum, and their movement on the window into the cochlea generate waves within the fluid-filled cochlea, and that causes the basilar membrane to vibrate. As the basilar membrane moves up and down in response to that, as you can see in this diagram, the hair cells will be bent where they touch the roof of the organ of Corti, and then as the basilar membrane moves downwards, the hair cells will straighten again. And just like with touching the hairs on your arm, that movement of the base of the hair is detected by the sensory receptors, by the neurons, and that generates the sensory response. In the next slide, what we can see is the group of stereocilia arising from a hair cell. So this is an electron micrograph showing a row of stereocilia from a group of hair cells. And as you can see, there's a group of stereocilia of different heights. The model that we see in the next slide shows how those inner hair cells might be activated. The tips of the stereocilia, the hairs arising from the basal cell, are linked by a structure, an actin-based structure called the tip link. So as the hair cells move, as you can see in the diagram, if they move in one direction, the tip link will be pulled, and the tip link connects with a mechanical gate on the next stereocilium. So as the hair cell moves, pulling on the tip link actually opens a mechanical channel. It's a mechanically gated channel that allows sensory transmission to occur. And this occurs because when the channel opens, we get depolarization of the hair cells, and that's detected and converted into a sensory signal. The next slide shows the auditory pathway that these sensory neurons take. So arising from the hair cells, we have a group of neurons that connect with the sensory pathways of the central part of the brain. So they pass to the brainstem, and then they're routed to the region of the thalamus. Again, if you remember, the thalamus is the gateway to the cortex, and this is the medial geniculate nucleus. So it's a slightly different nucleus to the one that vision goes to, which is the lateral geniculate nucleus. From the medial geniculate nucleus, the neurons pass to the auditory cortex, which is in a region of the brain called the temporal lobe. Just like the other cortices, the somatosensory cortex, the visual cortex, the auditory cortex is laid out as a map. And in this case, it's a frequency map of the auditory system. So at one end we have high frequencies, at the other end we have low frequencies. Those are detected within the sensory cortex, and then the higher regions of the cortical system process the sound to identify different characteristics, enabling us to differentiate, for example, between plain sounds, to identify speech, and to recognize different sounds. And it's that high-level processing, so for example, that allows us to identify the difference between a bird song and a phone ringing, or something like that. So this depends on the mechanical properties of the basilar membrane in the hair cells, and that initial processing that then allows us to aggregate together the different types of sensory input so that we can interpret them and recognize what the sounds are.